did the mice die? No, the mice did not die because our strain bacteria was avirulent, meaning it did not have the capability of causing diseases. The S strain bacteria, when it was subjected to heat, the high temperature, it denatured the polysaccharide, making it avirulent. And when injected into the mice lived. DNA is the one, directly or indirectly, the one that is causing the infection in the mice. So this is what they found out. Bacteriophages are nothing but the viruses that are capable of infecting bacteria. Hello everyone, a warm welcome to the session for class 12th biology. I'm Dr. Divya, biology faculty. So in this session of chapter 5, that is molecular basis of inheritance, we will learn about DNA as a genetic material. So in the previous session, we got to know about the properties of DNA, how DNA is wound inside the nucleus of a cell and all that. In this material, is DNA that is being transformed into another organism is what we will look into. So there are some experiments that has been conducted by few of the scientists who proved that it is the DNA that goes from one organism or transfers from one organism to another, and it is not the protein. So that is when they found out DNA is the one that acts as the genetic material that carries information from one organism to the other or from one cell to the other. So what were these experiments? We will look into. So transforming principle. So one of the experiment that was conducted in the year 1928 by a scientist by the name Frederick Griffith. So it's also called as Griffith's transforming principle or Griffith's experiment to prove DNA as a genetic material. So he conducted a series of experiments on the bacteria, that is Streptococcus pneumoniae. Streptococcus pneumoniae is a bacteria that actually causes pneumonia, and he witnessed a miraculous transformation in the bacteria. So this was an accidental discovery. Actually, he was working in his laboratory with few other microorganisms, and what he did was he forgot to clean the Petri plates and he left the Petri plates as such. The next day when he came, he saw that in the Petri plates he found some shiny molds of bacteria and all that were growing. So that is what made him curious to study about it. So during the course of his experiment, a living organism had changed in physical form. So the next day when he came back, he saw that the physical form of Streptococcus pneumonia had completely changed. So that curiosity led him to perform this particular experiment. So when Streptococcus pneumoniae bacteria are grown on culture plates, some produce smooth shiny colonies while the other produce rough colonies. So the Petri plates that he left without washing, when he observed it the next day, he found out same species of bacteria. It was Streptococcus pneumoniae itself, but there were two different colonies of Streptococcus pneumoniae, wherein one of the colony, the bacteria produced smooth shiny texture on their surface. They had smooth shiny texture, and other bacteria did not have this smooth shiny texture. So therefore he called those bacteria, one group of bacteria has smooth shiny colonies which he labeled as capital S, and the other group has rough colonies which he named it as capital R. So on the Petri plate, what did he find? He found groups of colonies of bacteria, wherein few of the bacteria, they were S, that is, they had a smooth shiny texture on their surface, and few colonies of bacteria had rough texture on their surface. So this is what he observed. Now he wanted to know why is it that way. So he found out that it is because the S strain bacteria, when I say S strain bacteria, what it means? It is the bacteria which is having a smooth shiny texture, right? Smooth shiny texture is actually because the bacteria is coated by a mucus covering, and that mucus is nothing but made up of polysaccharide, which is a sugar. So it is made up of a polysaccharide, and the rough colony bacteria, when he observed, he found out that this particular polysaccharide mucus coating was not present on them, and that is why they didn't have a shiny texture, and he called it as the rough strain bacteria or rough textured bacteria or R strain bacteria. So what he did was he wanted to find out the difference between these two bacteria, what each bacteria can do when it is injected into a mice. So what is the function of these two bacteria? It is to produce pneumonia, because what did he choose? He chose Streptococcus pneumonia bacteria, right? And he observed in one of the colonies of Streptococcus pneumonia, they had smooth coat and the other had a rough coat. So he conducted experiments on the mice. So what he did was he infected the mice with S strain bacteria. So S strain bacteria is virulent. Virulent meaning it has higher capability of causing disease. Which disease? Pneumonia. So it is called virulent S strain, and the mice died of pneumonia. Of course, because it is virulent, and if it is injected into the mice, within few days the mice will contract pneumonia and they will die. But again what he did was, for other group of mice, he infected or injected them with R strain bacteria. Now, did the mice die? No, the mice did not die, because R strain bacteria was avirulent, meaning it did not have the capability of causing diseases. S strain bacteria had mucus coating, that is polysaccharide coating around it. Therefore, it was virulent and capable of causing the disease, and that is why the mice died. But R strain bacteria, it did not have mucus coating around it. Therefore, it had a rough texture, and because of the absence of the polysaccharide coating, they were avirulent, that is, they were not capable of causing diseases, and hence the mice lived. So, this was his first experiment. Now, between the two colonies, he got to know the difference, right? S strain is virulent and R strain is avirulent. Virulence causes diseases. Avirulence, it is not capable of causing disease. He was not satisfied with this. This set of experiment was not just enough for him. So, what he did was, next, Griffith was able to kill the bacteria by heating them. So, he killed the bacteria by heating them. It is not necessary to kill the R strain bacteria, because obviously R strain bacteria is avirulent. You kill it or not, it will not be able to cause disease itself. So, which bacteria did he heat kill? He subjected the S strain bacteria to high temperature and he killed them. And he observed that the heat killed S strain bacteria. When he injected into the mice did not die. So, what did he do? He took the S strain bacteria, he subjected the S strain bacteria to extreme temperatures and he killed the bacteria. Therefore, it became what? Avirulent, right? And then he injected it into the mice and the mice lived. Why did the mice live? Because you would have learned this in your biomolecules chapter, in your class 11th of first PUC, right? Wherein proteins at higher temperature, they get denatured. So, here, what is this particular S strain bacteria or any sort of biomolecules for that matter, if subjected to high temperature, they can get denatured. So, here, the S strain bacteria, when it was subjected to heat, the high temperature, it denatured the polysaccharide, making it avirulent. And when he injected into the mice lived. Next, again, he was not satisfied with this particular experiment. So, he went in for another experiment. What he did was, now he mixed S strain bacteria. He took a syringe. In that, he mixed S strain bacteria along with R strain bacteria. Now, both of these are avirulent itself, right? Because avirulent means they are dead. So, and then he injected into the mice. This is quite interesting. What happened? The mice died. Now, you can ask me, why did the mice die? Because here, S strain and R strain he mixed. So, here what happened in S strain, the genetic material that is there in this particular bacteria, the DNA, it transformed and it entered into the live R strain bacteria. And now, because it has the genetic material and the genetic material is capable of causing pneumonia, because the S strain DNA will not get, you have studied about the properties of DNA in the previous session, right? Wherein we got to know that DNA cannot be broken or DNA will not get altered or the genetic material doesn't get altered or because it is not unstable. It is quite stable, right? So, therefore, whatever amount of chemicals or heat you subject it to, they remain stable. So, that is the reason why, though you subjected this to heat, the strain, and the bacteria died. In spite of that, their genetic material was intact. And when you mix that strain, heat-killed strain with R strain, that is live R strain, the genetic material got transferred, or it moved into the R strain, and from there it actually, when it was injected into the mice died, or because it contracted pneumonia. So therefore, from Griffith's experiment, we get to know that the DNA is the genetic material that is responsible for causing diseases as well. So whether the bacteria is dead or not. So the bacteria you can kill the bacteria because by denaturing its protein structures, right? By subjecting it to heat. But the genetic material never dies. So therefore, it's not the protein, or it is not the polysaccharide coat that causes the death of the mice, but it is the genetic material in the mice that carries the gene for pneumonia, or that genetic material in the mice, strain mice, is the one that actually made the mice to die. So this was his experiment. So he concluded that the R strain bacteria had somehow been transformed by the heat-killed strained bacteria. So some transforming principle is involved here. What happened? Transferred, that is, some transforming principle transferred from the heat-killed strain had enabled the R strain to synthesize a smooth polysaccharide coat. Now, in the DNA strand of the strain bacteria, what do they have? They have the gene that produces the polysaccharide coat. So, this is what happens. So, to tell you, we'll just brush up. So, strain bacteria, it has mucus coat. Mucus coat. What is that mucus coat made up of? Polysaccharide. And this polysaccharide is produced by a gene that is present on the DNA strand. Now, the R strain bacteria, it doesn't have the mucus coat. So, therefore, it is not capable of causing the infection. So, here what happened was, though this strain, when he heat killed it, and he mixed it with the R strain and he injected into the mice, this DNA or the genetic material from S strain got entered into the R strain, and it started producing what? It has entered into the R strain. So therefore, because it came from here to the R strain, it will carry the gene. What gene? The gene that is responsible for producing the polysaccharide. So therefore, this also started to produce, the R strain also started to produce the mucus coat. Therefore, it was capable of causing the infection. So therefore, DNA is the one, directly or indirectly, the one that is causing the infection in the mice. So this is what they found out. So therefore, he concluded that this might be due to the transfer of the genetic material from the S strain to the R strain. However, the biochemical nature of the genetic material was not studied in this particular experiment, but still his experiment proved that DNA is the one that carries the gene which is capable of killing this particular mice, wherein they can contract pneumonia and they can die. So this was one of the experiments. So what did we learn from this? DNA is, of course, the genetic material that is being transformed from one organism to another. Now, there's another experiment which was conducted by Hershey and Chase. So they also proved that DNA is the genetic material. So the unequivocal proof that DNA is the genetic material came from experiments of Alfred Hershey and Martha Chase in the year 1952. So Griffith's experiment proved that DNA is the genetic material, but biochemical studies was not done on that. But when Alfred Hershey and Martha Chase conducted the experiment in 1952, people all over the world, they believed that, or they agreed that DNA is the genetic material that moves from one organism to another. So they worked with viruses that infect bacteria. So all of you are very familiar with the word bacteriophages, right? So what are bacteriophages? Don't think it is a bacteria. Almost you get confused because there is the word bacterio in the beginning. Bacteriophages are nothing but the viruses that are capable of infecting bacteria. So for their experiments, so like how Griffith chose Streptococcus pneumoniae bacteria for his experiment, these people choose virus which infects the bacteria, that is bacteriophage for the experiment. So the bacteriophage actually it has the capability of attaching to the bacteria and then transferring its genetic material into the bacteria. So that is what they do. So if this is the bacterial cell, for example, these bacteriophages, they have tail fibers like this. So they have the genetic material. This is the protein coat. Inside the protein coat, they have the genetic material. So they will go and cling on to the cell wall or the surface of the bacteria, and then they will transfer their genetic material into the bacteria. So that is how the virus or the bacteriophage's genetic material enters into the bacteria. Using this, they actually proved that DNA is the genetic material. So we'll see what they did. So the bacterial cell, what will happen is, now I told you the bacteriophage will go and cling on to the surface of the bacteria and it will deliver the genetic material into the bacterial cell. Now the bacterial cell cannot reject it. It will treat the genetic material, virus DNA, as its own DNA, and it will start replicating and making more virus particles. So that is how viruses usually replicate, be it inside a bacteria or be it inside human body also. That is how they replicate. And Hershey and Chase worked to discover whether it was protein that is getting replicated. Replica means copies of it, multiple copies of it are being produced. Okay. So here they wanted to discover, okay, something is entering from the bacteriophage into the bacteria and it is replicating inside. That something, is it the DNA that is getting replicated or is it the protein coat that is getting replicated, is what they want to see. So they conducted the following experiment. So first what they did was, they took 2 Petri plates and they prepared a suitable media to grow viruses on it. Viruses, it is nothing but the bacteriophage that I'm talking about. So they grew some viruses on a medium that contained radioactive phosphorus. So media. So always a media is something which looks jelly-like and it solidifies because agar is added. That will be poured onto the Petri plate so that it provides nutrition to whatever you want to grow on it, especially a fungi, a bacteria or a virus. And the media, composition of the media varies depending on what organisms you choose for culturing. So here also they took a medium, they put viruses or bacteriophage in that, and to that medium they added something extra. What did they add? They added radioactive phosphorus. And to one media, to another media, in one Petri plate they added radioactive phosphorus, and to another Petri plate, one more Petri plate, they added radioactive sulfur. So 2 separate Petri plates. Okay. And viruses grown in the presence of radioactive phosphorus contained radioactive DNA. Why did it contain radioactive DNA? Because we have learnt in the previous session, what is the backbone of DNA made up of? It is made up of phosphorus and sugar molecule, right? So therefore, because phosphorus present. So because phosphorus present in that DNA became radioactive. Because what did we add here in one of the culture plate? We added radioactive phosphorus. That radioactive phosphorus went and binded with the phosphorus that is present in the DNA. Therefore, that phosphorus became radioactive. So therefore, the DNA in this, the DNA of the virus in this particular Petri plate, it became what? It became radioactive. Next, but in this particular Petri plate, the phosphorus did not become radioactive. Contained radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. Here in the virus, now I told you the virus contains a protein coat. So this is the protein coat of the virus. What it is made up of? It is made up of protein, and inside it is made up of the DNA, right? Now this protein coat, it contains sulfur. Sulfur is present, but in the protein coat, phosphorus is absent. In the DNA, sulfur is absent, phosphorus is present, right? So when we grew this particular bacteriophage or virus in a media containing radioactive phosphorus, then the DNA of the virus will become radioactive, but the protein will not become radioactive. And when we grow the same virus in a media containing radioactive sulfur, the DNA will not become radioactive because it doesn't contain sulfur, whereas the Protein coat will become radioactive because it contains sulfur. Now you got an idea of what it is, right? Similarly, viruses, it's the same thing that I explained. So moving further, radioactive phages were allowed to attach to E. coli. So we'll just brush up here. So what now? This is radioactive. So this bacteriophage is having radioactive 35S labeled protein capsule. So this was grown in sulfur media. This one was grown in sulfur media. This one was grown in phosphorus media. Because this was grown in sulfur media, the protein coat will have sulfur. So that is why they have colored it red. Okay, protein coat will have sulfur. Because this was grown in phosphorus media, the DNA will have phosphorus. So that is why DNA they have marked it as red. Okay, so for red it is sulfur, for black it is DNA. Now you can understand, right? Red it is sulfur, black is DNA there. So it is understood. Now then what they did was, in two separate experiments, this set of bacteriophages, they allowed it to grow along with bacteria. So there what happened? This went and clinged on to the bacteria. It went and clinged on to the bacteria. It released its genetic material, and after the release of the genetic material, so they did it in three steps. One is infection, the second one is blending, and the third one is centrifugation. So in infection is, now we have a petri dish. In that petri dish we have the bacteriophage or the virus whose protein coat is radioactive because it contains sulfur, and its DNA is not radioactive because it contains phosphorus, because we have grown this only on the sulfur media without phosphorus. So that's why. And next is infection stage, wherein both when they are growing, the bacteriophage will go and bind on to the cell wall of the bacteria, and once it clings, it will start releasing its genetic material into the bacteria. Now the DNA is inside the bacteria. DNA which is not radioactive, right? It's not radioactive because it cannot take up sulfur, doesn't have sulfur. This is radioactive. This protein coat is radioactive, but this is not radioactive. So then they blended, or they put it to centrifugation. Centrifugation is a technique wherein with high speed rotation, the material is subjected to high speed rotation so that the particles can get separated. Now the bacteria will separate and this radioactive protein coat will get separated. And when he observed these bacteria, he got to know that these bacteria contained the DNA inside it and not the protein coat. So that was one of the experiment. In the second set also he did the same thing. The bacteria bacteriophage virus which he grew in radioactive phosphorus. Now it contains radioactive phosphorus, but it doesn't contain radioactive DNA. It will contain, but it will not contain a radioactive protein coat, right? Because protein doesn't have phosphorus. It has sulfur, but DNA has phosphorus. So that's why. Then again, infection with the bacteria. Now they will deliver the genetic material. So this is radioactive. The DNA is radioactive. Then same thing, he'll go in for centrifugation, wherein radioactive phosphorus will be detected in the cells, and whatever protein coat, this is a protein coat, will come out, it will be not radioactive. So therefore, from these two experiments, they got to know that it is not the protein coat that enters into the bacteria, but it is the DNA that enters into the bacteria. Therefore, DNA is the genetic material that enters from the bacteriophage or the virus into the bacterial cell. So this experiment proved it. So radioactive phages were allowed to attach to E. coli bacteria, Escherichia coli bacteria. Then as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. So this was what they found out. It's the same thing in one slide itself. I've explained it to you. So therefore, they got to know that it is DNA, that is the genetic material that is being transferred from one organism to another. In this case, from the bacteriophage into the Escherichia coli bacteria. So these two experiments proved that DNA is the genetic material. So therefore, whatever organisms we study today, we get to know that the genetic material that can be transferred from one organism to another is the deoxyribonucleic acid, that is the DNA. So this was about this session. So in the coming session, we'll learn about the properties of the genetic material. That is, if a material has to be called as a genetic material, that is, if a DNA or a RNA has to be called as a genetic material, what are the properties it has to have within itself? And we'll study about the RNA world as well, because RNA is also the genetic material, though it is not a genetic material of eukaryotes, but still it is the genetic material of some of the prokaryotes, especially the viruses. So we have to learn about that as well. So see you in the next coming session with these two topics. Thank you.